Hi, so we'll start today's session and I'll share my screen and you can let me know if my screen is visible. I hope my screen is visible. So, good evening. Okay, one second. Okay, thank you. Yeah, so my screen is visible. Thank you. So, good evening, guys. My name is Aishi and today we are at the ninth week live session of this course Biochemistry taken by Professor K. Subramaniam from IIT Madras. And basically in these live sessions what we do is that we discuss some relevant questions, previous year assignment questions, and we try to find the answers to those questions, and we try to discuss the concepts behind those questions as well. So that is the main point of these live sessions, and today is the ninth week live session of this course, and we'll be discussing some of the previous year assignment questions. Okay, so if at any point my screen stops showing or if I become inaudible, please let me know by either unmuting yourself or by typing it in the chat box. I'll check and I'll reshare my screen. Okay, so moving on to the first question for today. Okay, so protein kinases are a group of enzymes that possess a catalytic subunit that transfers the gamma terminal phosphate from nucleoside triphosphates, often ATP, to one or more amino acid residues in a protein substrate side chain, resulting in a conformational change due to a covalently attached molecule affecting protein function. This type of inhibition belongs to which of the following classes? Competitive, non-competitive, allosteric, uncompetitive, irreversible. So I'll give you guys 1 or 2 minutes if you can try answering this question, and then we'll move ahead to discuss the question and to discuss the concept behind the question. Anyone? Okay, so the answer will be allosteric. So the question says protein kinases are a group of enzymes that possess a catalytic subunit that transfers the gamma terminal phosphate from nucleoside triphosphates to one or more amino acids residues in a protein substrate side chain, resulting in a conformational change due to a covalently attached molecule affecting protein function. So what does this type of inhibition, which class does this belong to? So we'll basically talk about the thermodynamics and the kinetics of how what thermodynamics of a reaction says. It basically says whether a reaction will occur, and the kinetics says when the reaction will occur. Now, what governs when the reaction will occur? So for a reaction to occur, it needs to destabilize the existing state, input energy. So there is a huge activation barrier that needs to be crossed basically for the reaction to occur. And for reactants must find each other, they should meet in proper orientation and hit with sufficient force for the reaction to take place. Okay, so productive collision will lead to the enzyme substrate complex for the reaction for the basically to accelerate to basically promote the reaction, but there can be many non-productive collisions where the reactants are colliding in non-compatible orientations which do not ultimately lead to the reaction product formation. So for what are the needs of typical chemical reactions? We need large number of molecular collisions definitely, and we need the collisions to happen more violently enough to break pre-existing bond. They should not bounce back. They should collide violently enough to break the pre-existing bonds. And they need high concentration to find each other at significant rate. Okay. It's the energy profile of a hexagonic reaction, reactants, transition state, and the products. And this is EA is known as the activation energy of the reaction. Okay. So the x-axis talks about the progress of the reaction, the y-axis talks about the free energy. Here, the x-axis talk about the energy per molecule, while the y-axis will talk about the number of molecules. Okay, so here the molecules with average energy. Here will be the many molecules that have enough energy to undergo the enzyme catalyst chemical reaction. Here, there's almost no molecules have the very high energy needed to undergo and catalyze chemical reaction. Okay. Whereas in case of temperature 2, if you see the bell curve, the majority of the molecules are in this range where many molecules have enough energy to undergo the enzyme catalyzed reaction. Okay, this is the bell-shaped curve. So now the molecules with sufficient energy to cause the reaction will be 40%, which is previously even less than 5%. Okay. Now what are the roles of enzymes? Enzymes basically make reactions easier to occur at reasonable temperature by lowering the activation energy of the reaction. Enzymes are nothing but biological catalysts. They neither get used up nor get consumed in the process. What they do is that they lower the activation energy of the reaction. So this is the energy necessary to overcome the status quo. Thermodynamic favorability, and this is the ease of initiating reaction. So if you see the reactants to products, this is the energy released by the reaction, and this is the activation energy that is needed to be crossed in order to form the product. This is the activation energy without the enzyme. This is the energy with the enzyme. So you see the enzymes have lowered the activation energy. So now it's easier for the reactants to get converted to the products. So as I said, enzymes are biological catalysts. They promote a specific reaction but are not consumed in the process. So when we say promotes, they do not alter what would normally occur thermodynamically. They promote only one reaction only between specific reactants to give specific products, and they are reusable, that is they get regenerated in the process. So enzymes are biological catalysts. They are usually proteins. Some RNAs can also be catalysts. Sometimes RNA or RNA protein complexes can also act as catalysts. Okay. So basically, enzymes work as catalysts. We are providing an easy path to the same point. If you can see, this is the hard path, but enzymes will help you get an easier path. Now, how do enzymes do it? Okay. So enzymes have binding affinity for their reactants, that is substrates. Okay. So if you see, this is the enzyme, and this will be, so basically by this, if it can bind to the reactants, it basically ensures that the reactants will be in very close proximity. So it is basically promoting the reactants in coming in contact with each other and in proper orientation so that the reaction can move forward. Okay. So enzymes bind only specific things, and they bind only in a specific 3D orientation. So specificity is the key to enzyme action. Specificity is very important in biological systems, and specificity is the key to enzyme action. Now enzymes can also orient substrates, always in productive orientation. As I said, huge number of collisions will not matter if the collisions are not made in proper orientation. So what enzymes do is that they orient substrates always in productive orientation. You can see this is a productive orientation. These are many non-productive collisions. Okay. Now enzymes can cause bond strain. Basically, it can destabilize the existing bonds. Okay. So it will cause some kind of physical and chemical strain to the reactants, and it will cause destabilization of the existing bonds. Now what is the active site? The active site is the region on the enzyme where the substrate binds. Okay. Now what is the induced fit of a substrate? Okay. That is, the substrate will bind and the enzyme will modify its cavity or its site in such a way so as to properly bind to the substrate. Okay. So enzyme substrate interactions have been explained by two popular models, the Fisher's lock and key model or Koshland's induced feed model. Okay. So this lock and key model, this is the active site, this is the substrate. The substrate will come in contact with the enzyme active site and it will give rise to the products. For the Koshland induced feed, the enzyme might not be compatible, exactly complementary structurally to the active side of the enzymes, but once it gets in touch, there's a distorted bond formation. It basically gets modified so as to accommodate the substrate properly, and the products are then released. So what is the catalytic cycle of an enzyme? Number one, substrates will enter the active site. Enzyme will change shape, so its active site embraces the substrates. That's what I was talking about in induced feed. So what happens? Substrates will enter the active site. The enzyme will modify its shape a little. So then the active site will embrace the substrate, and that will be induced feed. Substrates held in active site by weak interactions such as hydrogen bonds and ionic bonds, that will be enzyme substrate complex. Then the active site and R groups of its amino acids can lower the activation energy and speed up a reaction by acting as a template for substrate orientation, stressing the substrates and stabilizing the transition state, or providing a favorable microenvironment, or participating directly in the catalytic reaction. This is how the active site of the enzymes can basically lower the activation energy and speed accelerated reaction. Excuse me. Then fourth step is substrates are then converted into the products. Okay, and then the products are released. Okay, and then the active site is available for the two new substrate modes, and then basically the entire cycle starts again, resumes again. Now, what is chemical bond strain? Basically teasing the bond to fall apart. Okay, so if you see over here, there is no catalysis, there is acid catalysis over here, there is base catalysis over here, and there is both acid and base catalysis, which happens very fast. So any kind of chemical strain will basically pull the existing, will basically pressurize the existing bonds to fall apart. Okay, so that is what we call the chemical bond strain. Now there is an important role of cofactors as well. What are cofactors or coenzymes? Cofactors are basically non-polypeptide or non-protein things at the active site that will help enzymes do their job. So apart from enzymes, there is another component, cofactors or coenzymes, which basically help the enzymes do their job. So cofactors are non-protein enzyme helpers. Example like zinc, okay, zinc acid and everything. And the coenzymes are basically organic cofactors. So enzymes partake in reactions but are not consumed in them. So as I said, enzymes will participate in the reaction. Basically, it will increase the. What it will do is it will accelerate the reaction, but it will not get consumed up in the process. This is the enzyme. This is the active site. The molecule A is going. Then this is the complex is being formed. Catalysis happens, and then enzyme gets released and molecule B product is released. So it can convert many substrates into products. Okay. If we talk about a particular enzyme, let's talk about lysozyme. It's basically an enzyme that works at low pH, like pH 4, 5, acidic kind of acidic pH. Why? So basically it works on its substrates in a manner which is being promoted by the acidic environment. Okay, so that has all to do with the reaction mechanism that you see over here. So that is why lysozyme shows its optimal activity at pH 4 or 4 to 5. So in summary, enzymes are basically molecules which will bring reactants and substances in close proximity. They'll align substrates in proper orientation. They can act as a lever. Basically, they can do small change shape and translate to large force, and can release products when reaction is done. It can rebind more substrates, and there are many small steps, each easily achieved rather than one huge leap. Okay. Now, enzymes carry out reactions in a series of small steps rather than one energetic step, as we said. Okay. So the reaction rate will depend. If we put substrate concentration on the x-axis and rate or velocity on the y-axis, we discussed the Michaelis Menten curve in the last class. It will basically show this type of curve. So an enzyme catalyzed reaction can be saturated basically. So with increasing substrate concentration, the rate or velocity will also increase until it saturates or it reaches a plateau. That is the maximum velocity or Vmax. And the half Vmax will correspond to the Km or the substrate affinity, which is basically explained in the terms of substrate concentration. So the lower the Km, the better the enzyme recognizes substrate, finds it at low concentration. So if your Km, which comes in the form of substrate concentration, is very low, binding affinity, that is the lower if the Km is very low, that is the enzyme is very good and recognizing substrate even with at lower concentrations. But if your Km is high, that is your binding affinity, then your enzyme will take a lot of substrate to be able to recognize that. The higher the Vmax, the more substrate an enzyme can process per minute. So that is the significance of what the Vmax stands for. The maximum velocity stands for the more substrate and basically stands for this, and the higher the Vmax, the more substrate will an enzyme can process per minute if substrate is around. Now what are the things that have, what are the factors that are involved in the activity? Okay, something that can affect the protein structure and also affect the enzyme activity. Temperature and pH are the foremost conditions. So each and every enzyme has a proper temperature range in which it is the most active. Okay. So for example, this range is most popular for optimal. It's popular as an optimal temperature for human enzyme, because this is the relevant physiological temperature of the human body. Whereas this temperature, which is very high, is the optimal temperature for enzymes of thermophilic bacteria, because that is the optimum temperature for tolerant bacteria. Similarly, for pH, the rate of the reaction can also depend on this factor of pH. So the pH optimum of pepsin is quite low. It's in the acidic range because pepsin is found in the gastric juice. So that is where it works. And the pH optimum of trypsin is quite high in the alkaline range. Now, enzyme regulation can be activity controlled and continually adjusted. Okay. So, what are the principal ways of regulating enzymes? Competitive inhibition, allosteric inhibition, or covalent modification, that is phosphorylation. So, what is competitive inhibition and what are competitive inhibitors? So, if you can see, the name is pretty self-explanatory. So, what happens? Your substrate will bind to the enzyme at the active site and that will be the first step for your product formation to occur. But what if the enzyme, the substrate is not able to attach to the enzyme only? And how will that happen when the active site is already being occupied by someone else? Okay, think of it like how you occupy seats in the bus. So if you get on a bus and if you find a seat empty, that's fine, because you can go and sit and sit on that chair. But if you enter the bus, board the bus, and you find that it is already being occupied by some other person, you cannot basically push that person to sit there. So that is how the competitive inhibitors work. So when the enzyme and substrate come into contact, the substrate can all find that there is already something already occupying the active site of the enzyme. Okay, so the whatever was occupying the active site of the enzyme will be called now competitive inhibitors. Okay, so basically what they do is that they bind to active site unproductively and block the true substrates from accessing the active site. So they are stopping the reaction, because now the substrate cannot access the active site. The reactions needed will not be able to occur, and because there is inhibition by the competitive inhibitors. Okay, so S means substrate, I means inhibitor. They bind to the same site in case of competitive inhibition. So if you see, this was the site of the substrate 1, substrate 2, but I, which is the inhibitor, is always already occupying this site. So the substrates cannot bind. Now competitive inhibition happens like this: the active site of the enzyme, the substrate will come attached to this enzyme, then products are formed. But here in this case, the inhibitor will bind to the active site, substrate will not be able to bind, and hence no product formation. So inhibitor and substrate both bind to the active site of the enzyme, and binding of an inhibitor will prevent the substrate from binding. Okay. Now what are allosteric inhibitors? Allosteric inhibitor basically means other side. So whatever happens, it distorts the conformation of the enzyme. Okay. So this is allosteric inhibitor. This is the enzyme. This is the place for the substrate. Okay. This is a negative allosteric inhibitor, for example. So the substrate is attached. Now the allosteric inhibitor is attaching to not the active site but to another site on the enzyme. But because of this attachment, the enzyme now modifies the structure of its active site. So now it is no longer compatible with the substrate. So it basically distorts the conformation of the enzyme, which is why the substrate is given off and the enzyme does not, and the reaction does not take place. See, this is the allosteric site, this is the active site. Substrate is attached to the active site. Okay, product formation will happen. But if there is an allosteric inhibitor, it will bind to the allosteric site, not to the same site. So this is not competitive inhibition. But now the structure, the size of the active site has now changed because of the allosteric site occupation by the allosteric inhibitor. So now the reaction will not occur. Okay. So that was all about negative allosteric regulators or inhibitors. What are positive allosteric regulators? This is something which helps the enzyme in doing their job. So you see, here the enzyme and the substrate were not compatible with each other. See, they're not really compatible with each other, so they're not coming in contact. This is an allosteric site here. Now if a positive allosteric activator comes and sits here, it changes, modifies the enzyme structure, active site structure, so that now it can bind to the substrate. So the positive allosteric regulators basically helps enzymes to work better. It promotes or stabilizes an active conformation. You see this image, there is little or no product formation because the active site is not really compatible with the substrate. But now the allosteric site activator has gone attached to the allosteric site. Now it has distorted the structure of the enzyme to make it suitable for substrate attachment. So allosteric regulators can change the shape of curve of the enzyme. Okay, so if you see, this is allosteric enzyme with 4 subunits, the active site 1 of 4. The allosteric activator will stabilize the active form. Okay, and then so that is what is happening. There is an oscillation between the active and the inactive form. The inhibitor, this is a non-functional active site because the active site is no longer compatible with the substrate. Okay. Another frequent regulatory modification is phosphorylation of enzymes. Okay. You see the image over here. Protein kinase basically adds a phosphate group, and protein phosphatase basically removes the phosphate group. So suppose this enzyme was glycogen phosphorylase B. This is the inactive form. When there is a kinase activity, there is phosphate group attachment which makes it an active form. Whereas if you want it to go back to the inactive form, it will be the job of the phosphatase to make it go back to the inactive form. So in summary, all enzymes are catalysts, lower activation energy. Their mechanism of action we have discussed. Their enzyme kinetics, Vmax and Km we have discussed. And regulation of enzyme activity, competitive allosteric phosphorylation. So enzyme are key to life. So no enzyme will basically mean no life. Okay. So we have talked about the enzyme active site. Okay. And how enzymes can basically speed up reaction rate by lowering activation energy without being affected by the reaction it catalyzes. Okay. Now, enzymes are protein in nature. That's all enzymes are not protein in nature. There are some RNAs also that can act as enzymes. So globular proteins can be enzymes. They are ribosomes are RNA molecules with enzymatic activity. Then catalytic behavior of any enzyme depends upon its primary, secondary, tertiary and quaternary structure. Then enzymes of digestive tract and those found in blood are present in active form called zymogen or proenzymes. This is active site. So enzymes are composed of long chains of amino acids that are folded into very specific three-dimensional shape which contains an active site. Now the active site is a region on the surface of an enzyme to which substrates will bind and catalyze a chemical reaction. So enzymes are highly specific for the type of reaction they catalyze and for their substrate. And enzymes with higher specificity and accuracy are involved in copying and expression of genome. These enzymes have proofreading capabilities such as DNA polymerase. So why are the enzymes higher specificity and accuracy are involved in copying and expression of genome? Because if a mistake happens at that level, that will be life threatening. Okay, so the copying and expression of genome should be done perfectly, and that's why enzymes with highest specificity and accuracy are involved in this process. Some enzymes require non-protein molecule for their activity. So enzymes are conjugated proteins known as hollow enzymes. Hollow enzyme will be apoenzyme, which is the main protein part, and cofactor or coenzyme, which can be the organic or the inorganic part. Now, enzymes without non-protein part is inactive and called apoenzyme. And cofactors can either be organic or inorganic in nature. I have talked about mechanism of enzyme action in one of the previous classes. Basically, the enzymatic reactions takes place by binding of the substrate with the active site of the enzyme molecule by several weak bonds, and the formation of the ES necessary complex is the first step in the enzyme catalyst reaction. Then ES complex is subsequently converted to product and free enzyme. So we have talked about the lock and key model. We have talked about the induced feed model as well. So what are the factors that can affect reaction velocity? Temperature, obviously. pH we have discussed. These two we have discussed. Substrate concentration met also, because greater the concentration, better will be the outcome. Enzyme concentration, definitely products of the reaction, presence of activity inhibitor, allosteric effects, time, etc. So we have talked about the effect of temperature. There will be an optimum temperature for each catalyst. If this is a reaction velocity and temperature, they should be making people should be learning more about the optimal range of the enzyme before optimal temperature range of the enzyme before moving ahead with their studies. Effect of pH also shows different structures. So here also, since pepsin is found in gastric juice, they are basically they can basically perform the maximum to the maximum limit in acidic medium, whereas the opposite for trypsin. So rate of the reaction or velocity is directly proportional to the enzyme concentration when sufficient substrate is present, and accumulation of product in the reaction causes the inhibition of this enzyme activity. A study of reaction rate and how the changing response to change in experimental parameter is what we call kinetics. The amount of substrate present in one of the key factors affecting the ratio of reaction catalyzed by an enzyme in vitro. We have talked about this certain curve. We talked about what are the steady state assumptions we need to keep in mind before we move ahead with the Michaelis Menten reaction. Equation, sorry. So this is a mathematical equation that defines the quantitative relationship between the rate of an enzyme catalyzed reaction. with the help of this equation. So V naught is the observed velocity at the given substrate concentration. Km is the microspherin constant, and V max is the V naught is the observed velocity. V max is the maximum velocity at saturating concentration. Now, what is line view plot? Is basically a linear representation of Michaelis maintained. So double reciprocal bond. Both sides have been made reciprocal, and it's a linear representation and is more accurate and convenient for determining Vmax and Km. This equation is obtained by taking reciprocal of both sides of Michaelis Menten equation. Okay. So see, this is how the line format of Michaelis Menten will look like. It's a line viewer block plot, double reciprocal. So the first equation we were talking about is V naught equal to V max S by Km plus S. But now if we do do double reciprocal of the equation, this is what the equation stands out to be: 1 by V naught, 1 by S. Enzyme inhibition. Any substance that can diminish the velocity of the enzyme is catalyzed, and it include drugs, antibiotics, poisons, and anti-metabolites. It's useful in understanding the sequence of enzyme catalyzed reactions, metabolic regulation, studying the mechanism of cell toxicity produced by toxicants, and forms the basis of drug testing, drug designing. We have talked about competitive inhibition. We will also talk about the remaining types of inhibitions. So in competitive inhibition, we have talked exactly how it happens. So substrate is about to get attached to the enzyme, but an inhibitor is always there already there in place, and so this competition between the substrate and the inhibitor, and the inhibitor is already there on the active site. So that is known as competitive inhibition. Anyone who has not understood the meaning of competitive inhibition? Okay. For other case of non-competitive inhibition, so here in case of this, we can see the inhibitor can attach to the enzyme or to the enzyme substrate complex, and after that no reaction will be taken forward. So that is basically the schematic of a non-competitive inhibition. Then we come to uncompetitive inhibition. It binds only the instancer complex. It does not have the capacity to bind to the more free enzymes. Not overcome by increasing substrate concentrations, and both the Km and Vmax are reduced. This is what the uncompetitive inhibition looks like. Okay, so this is the enzyme and this is a complex, and then the ESI complex is also formed at the same time. We have talked about competitive inhibitors. So these are molecules that are similar in shape to the substrate, structural complementarity with the active side of the enzyme. They bind to the enzyme's active site, preventing the real substrate from binding. This molecule competes with the substrate for the active site of the enzyme and this can be overcome by increasing substrate concentration. This is competitive inhibition. Now that the substrate cannot get to the active site because it is blocked by the inhibitor. So Prontosil is a competitive inhibitor. It's nothing but an antibacterial drug. So bacteria, what happens is that they require folic acid for replication of the genetic material. So what happens? Prontosil binds to the enzyme that makes folic acid, preventing other substrates from binding. And as a result, folic acid is no longer made and the bacterial cell dies. And since animal cells don't make folic acid themselves, they do not have this enzyme, and so Prontosil has no effect on them. Now coming to non-competitive inhibition, it's basically when a molecule binds to the enzyme at a location other than the active site, and this binding alters the shape of the enzyme, changing the shape of the active site. The enzyme is now dysfunctional because substrate now cannot bind to the active site. Adding more substrate will not affect the reaction because the active site will still be unavailable. So we talk about cytochrome C oxidase that has a non-competitive inhibition. So it speeds up the reduction of oxygen to water in a cellular respiration. So without it, the reaction will not occur fast enough, and the organism will die because not enough energy is being released. For example, in case of cytochrome C oxidase, which follows non-competitive inhibition, the CN group will come and attach to the SH groups in the enzyme. This destroys the sulfide bridges and this claiming and this changing the tertiary structure of the enzyme. Change in the shape results in the change in the active site. Thus, the substrate cannot bind in chromosome. Cytochrome C oxidase is now functional. So, in competitive inhibition, where there is more substrate than inhibitor, the max rate of reaction can be reached. However, it takes a larger substrate concentration to reach maximum rate of reaction. Then coming to allosteric regulation, some enzymes have receptor sites away from the active site known as allosteric sites, and these enzymes are usually proteins made up of several subunits each with an active site, and substances that can bind to the allosteric sites may inhibit or stimulate uh, the enzyme activity. So allosteric regulation occurs. We can bring an activated to an allosteric site, stabilizes the protein conformation. This gives all the active sites available for the substrates to bind to them. Uh, for allosteric regulation, the binding of an activator or an inhibitor will affect the activity of all the active sites in the enzyme. You can see this active site substrate is sitting, but there is an allosteric inhibitor or allosteric activator which is bind here and it will change the entire trajectory of the inhibition. Anyone has any questions till now, or we'll move ahead? I'll give you 1 to 2 minutes. Okay. So in case of metabolic pathways, there are changes in cycles of enzyme catalyst reactions, right? So the metabolic pathways in our system is very complex. So there are cascading reactions that will happen. Correct. So most chemical reactions do not happen in one large step, but rather as a sequence of smaller steps or reactions. Okay. So the product of the first enzyme catalyst reaction is the substrate for the second enzyme catalyst reaction. That is how it will happen. So product of enzyme 1, substrate of enzyme 2. Product of enzyme 2 will be the substrate of enzyme 3. If you see the schematic over here, that is how it happens in majority of the cascading reactions. So metabolic reactions can either be a chain of reactions in a cycle. In a cycle, the end product is substrate for the first enzyme and it will start the pathway again. So if you see substrate 2, substrate 3 to product. Another type of inhibition is feedback inhibition. So basically it is a method of controlling metabolic pathways. So negative feedback inhibition is like a thermostat. When it is cold, the thermostat turns on the heater to produce heat. When it is too warm, the heat will cause the thermostat to turn off the heater. So that is how the feeding inhibition occurs. So heat has a negative effect on the thermostat and a buildup of product inhibits the enzymes of the reaction. It is negative feedback. Now many enzymatic pathways are regulated by feedback inhibition. At the enzyme's end product accumulates, it inhibits the enzyme by binding to the first enzyme in the pathway. This shuts down the entire sequence. Okay. There's a lot of roles of all these different products at different stages in the feedback inhibition. So if you see this is an example, through a series of 5 reactions, the amino acid threonine is being converted to isoleucine. As the concentration of isoleucine builds up, it binds to the allosteric site of the first enzyme. It is threonine deaminase, acting as a non-competitive inhibitor. So now there is too much of isoleucine built up in the body. So the body has to switch off the system by basically binding as an inhibitor to the first enzyme in the pathway. So that is also what is called feedback inhibition. Substrate is threonine. There are so many steps that will ultimately give rise to the end product. Now when the end product is accumulating, gathering up too much, it will go and bind to the allosteric site of the enzyme, first enzyme, feedback inhibition. Now the active site of enzyme 1 is no longer able to catalyze this conversion of threonine to intermediate A pathway switched out. As the product is used over time, the concentration of the product decreases and the inhibition product will detach from the enzyme, allowing it to become active once again and product form reduced product. And when the product concentration gets too high again, the product will once again allosterically inhibit the enzyme. Okay, so that is all about feedback inhibition. Move on to the next question. This question is done. We have discussed all the different types of inhibition, allosteric, feedback inhibition, all of that. Competitive, uncompetitive, non-competitive. We'll move on to the next question for today. I'll give you 1 or 2 minutes to wrap up the thoughts on this question, and if you have any doubts, you can ask me on the chat. I'll try to answer those. Then we'll move on to the next question.
Okay, so that is all about inhibition topic. We'll take a break for 5 minutes and we'll move on to the next question. Okay, so we'll move on to the next question for today. Next question is rhamnose is a naturally occurring deoxy sugar that is unusually found in the L form. Rhamnose is commonly found to other sugars in nature. It is a common glycol component of glycosides from many plants. Rhamnose is also a component of the outer cell membrane of acid-fast bacteria in the Mycobacterium genus, which includes the organism that causes tuberculosis. It can be considered a deoxymannose. How many carbons are present in its backbone? There are 4, 5, 9, 6, 7, 8. These are the options. I'll give you 2 minutes, 2 to 5 minutes to discuss, to see if you can know the answer, then we'll discuss. Okay. So the correct answer for this question will be option 6. So if we talk about glycosides, glycosides are nothing but the molecules in which a sugar part is bound to some other non-sugar part. Okay. So glycosides play numerous important roles in living organisms. That's nothing extra to say. That's true. Can't store important chemicals in the form of inactive glycosides. And if these chemicals are needed, the glycosides are brought in contact with water and an enzyme, and the sugar part is broken off, making the enzyme chemical available for use. Many such plant glycosides are used as medications. Okay. So formally what happens, a glycoside is any molecule in which a sugar group is bonded through its anomeric carbon to another group via glycosidic bond. The sugar group is known as the glycone part and the non-sugar group is the glycone and genin part of the glycoside. The glycone part can consist of several single sugar groups like monosaccharide or several sugar groups to oligosaccharide. So sugars found in glycosides may be monosaccharides like glucose, rhamnose, fructose or deoxysugars, which are more rare, like cimarose. I don't know exactly how to pronounce this. Cimarose found in the cardiac glycosides. So what are the terms used to describe glycosides? A glycoside is a glucoside because it has a glucose as the sugar component. Can be a pentoside, which is a sugar as a arabinose. Rhamnosides, which will contain rhamnoglucosides, which will contain both rhamnose and glucose. So a glycosidic bond is a certain type of functional group that joins alcoholic group of a carbohydrate molecule to an glycone molecule. So a substance containing a glycosidic bond is a glycoside. This is a glycosidic bond. Anomeric carbon glycosidic bond. Classification of glycosides. Glycosides can be classified by the nature of the glycone, the number of sugars, the nature of the glycoside, the botanical source, the therapeutic use, the type of glycosidic bond, the glycosidal linkage, and the chemical nature of a glycone. These are the various ways through which you can classify glycosides. Okay, so if we classify by glycones, so if the glycone group of a glycoside is glucose, then the molecule will be called a glucoside. If it is fructose, then the molecule will be called a fructoside, and if it is glucuronic acid, then the molecule will be called glucuronide. So these are the different sugars. So if it's a monosaccharide, one sugar, the biocides will be two sugars, triosides, three sugars, diocides, and the nature of the glycosides. So primary glycosides are originally present in the plant, such as purpura A. Secondary glycosides will result from removal of one sugar from the primary glycosides. So that proper example can be digitoxin. Now, if we categorize by type of glycosidic bond, then depending on whether the glycosidic bond lies above or below the plane of the cyclic sugar molecule, glycosides can be classified as alpha glycosides or beta glycosides. So these are the difference between the alpha and the beta glycosides. As you can see, alpha methyl glucoside, beta methyl glucoside, alpha methyl. So if you see, the only difference between these two is the basically the orientation of this. Okay, this is alpha glucose. So if the plane, so if the glycosidic bond is above the plane or below the plane, that will determine whether it's an alpha methyl glucoside and the beta methyl glucoside. You see, everything else is same, only this below and above the plane feature is different for these two, alpha and beta methyl glucoside. Okay. Now, in case of carbohydrates, we have the monosaccharides derivatives such as So these make sugar acids or acid sugars. So oxidation of an aldehyde ketone to acid, reaction from the C1 of aldose named as onic acids, and these sugars can reduce copper 2 plus to copper via Filling's or Benedict's test or Tollens test, silver plus to silver. A prime example would be gluconic acid. Then we have a reaction of the Benedict's test. Okay, so you can figure out the purpose of reducing sugars will basically give red PPT. So the copper will be oxidized, and the sugar will be copper will be reduced, and the sugar will be oxidized. So the sugar will be called reducing sugar, and reducing sugars will have a free anomeric carbon. Name monosaccharides derivatives. These make sugar acids or acid sugars. So you can get colorimetric glucose analysis. So enzymatic methods are used to quantify reducing sugars such as glucose. The enzyme glucose oxidase will catalyze the conversion of glucose to glucono lambda lactone and hydrogen peroxide. Hydrogen peroxide will oxidize organic molecules into highly colored compounds, and concentrations of such compounds is measured colorimetrically. Okay, so electrochemical detection is used in portable glucose sensors. To talk about monosaccharides derivatives, so these make sugar acids or acid sugars. I've talked about this. So oxidation of alcohol to acid with 4 electron loss can happen through reaction of C6 groups. Like C1 oxidation normally can happen on add losses, so that will be named as uronic acid, like uranate for conjugate base. So many L sugars are uronic acids. If you see beta D-glucuronate. On the other hand, monosaccharides derivatives, so these maybe make sugar alcohols or deoxy sugars. So it's basically reduction of aldehyde or ketone to alcohol. Only carbon not already in alcohol is the anomeric carbon C1, C2. Okay, and reduction of alcohol carbon to methyl or methylene. Okay, so you can react at any except the anomeric carbon. Many are L sugars and have specific trivial names. Okay. So the question was about rhamnose. We have already discussed about rhamnose. We'll move on to the next question, which is basically this one. Which of the following disaccharides is non-reducing? Trehalose, cellobiose, maltose, lactose, sucrose. Anyone wants to answer the question? I'll give you 2 to 5 minutes to think about it. Okay, so the answer will be trehalose and sucrose. So if we start from the basics, we have already talked about carbohydrates. What are carbohydrates? Are basically are polyhydroxyaldehydes or ketones or substances that yield such compounds on hydrolysis. They have structural roles such as cellulose in plants. They have energy storage roles such as starch in plant, glycogen in animals and bacteria. The classification of carbohydrates can be done as monosaccharides, simple sugar. The triose will be 3 carbons, so glyceraldehyde, tetrose will be 4 carbon erythrose, pentose will be 5 carbon arabinose, hexose 6 carbon aldose will be glucose, and ketose will be fructose. Then we have disaccharides, 2 molecules reducing sugar. It will have a free aldehyde or ketone group like maltose or lactose. Non-reducing sugar that will lack a free aldehyde or ketone group such as sucrose or polysaccharides. We have oligosaccharides. There will be 2 to 9 molecules basically of monosaccharides. For oligosaccharides, and for polysaccharides there will be 10 plus molecules like starch, dextrin, glycogen, cellulose, etc. Now, what is the effect of acid on carbohydrates? Acid or heat will convert disaccharides or polysaccharides to the monosaccharide form. And then if there are this loss of 3 water molecules, it will basically give rise to ring formation, sulfonyl derivatives, and whose phenolic derivatives will give colored complexes. So if we talk about some of the tests specific for carbohydrates, we have a Molish test, general test for all carbohydrates. The compounds basically give sulfonyl derivatives in the presence of H2SO4 concentrated. Then they react with alpha naphthol to give a purple ring. Okay, so we have glucose basically after in the presence of H2SO4 concentrated and release of 3 hydrogen water molecules, you give rise to sulfonyl derivatives, and then alpha naphthol will turn that into a purple ring. We have Benedict's test. It distinguishes between reducing and non-reducing sugars. So reducing sugars will have free aldehyde or ketone group. Basically, if you heat a mixture of Benedict reagent with reducing sugar in basic condition, it will lead to reduce CO2 plus to Cu plus as CO2 as yellow and then red color. Basically, it will give red color reducing sugar. Sugar plus Benedict will give carboxylic and copper oxides. Okay, the negative will stay this color and the positive will get red color. Then there is a test called Barfoed test. Basically distinguishes between mono and di sugars. So you heat a mixture of Barfoed reagent with monosaccharides in acidic condition. It will lead to that will lead to reduction of C2 plus to Cu as CO2O red precipitate. That will be the mono sugar basically. This is red precipitate is your positive reaction. This negative reaction is the blue color. Then there is a Biles test, which basically distinguishes between pentose and hexose sugars. The compounds basically will give, as we discussed before, will give rise to sulfonyl derivatives in the presence of concentrated HCl, and then when react with orsinol to give a blue-green pentose color. So this is the positive reaction. This is what happens when the reaction is negative. Then there is Sally-Warnoff's test, which basically distinguishes between ketose and aldose sugars. Compounds will give sulfonyl derivatives in the presence of HCl concentration, then react with isoorcinol to give orange color. So the ketose forms will give orange color. So if you see over here, there is a ketose form that will give an orange color if the test is positive. For example, there is fructose over here, it's a ketose form that will give orange color. If it's not positive, it's a negative one, will give a pale yellow color. Then we have iodine's test. Iodine's test basically is the test for polysaccharides. Okay, so there is an amylose which has a linear chain with I2 will give helically coiled polysaccharide chain and iodine. Okay, so as we know, I2 or iodine is an indicator of starch. Okay, so as you can see, I2 will form a complex with amylose, which is a component of starch, and the positive reaction will give rise to blue color. The positive is blue starch. So these are the carbohydrate tests that we have talked about: Molish test, Benedict's test, Barford test, Heligonov's test, Bial's test, and iodine test. So there's a way of moving ahead to figure out which unknown carbohydrate you have. So this schematic basically talks about that, in which order you should move on, in which order you should do the tests in order to find the proper carbohydrate that is present in the solution. If there is a network problem, 1 second, I'll just unshare for a minute. There's something wrong happening. Give me 5 minutes, I'll unshare and reshare. There's some network issue. Yeah, I reshare my screen. 1 second. Yeah. So this is a schematic that shows the exact order of tests that one needs to perform to figure out the unknown carbohydrate. Okay, carbohydrate. So classification of carbohydrates can be done on the basis of number of carbohydrate units. Monosaccharides will be 1 unit, simple carbohydrates.
Disaccharides will be 2 carbohydrate units, complex, and trisaccharides 3, polysaccharides many carbohydrate units. Position of carbonyl group can also be a determining factor for the classification. If it's at C1, the carbonyl group is an aldehyde, aldose. At any other carbon, carbonyl will be a ketone, which is a ketose. Like how we differentiate between glucose and fructose. Then number of carbons. So 3 carbons will give rise to triose, 6 carbons will give rise to hexose, 4 carbons will give rise to tetrose, 7 carbons heptose, 5 carbons pentose, and etc. Then obviously cyclic form will also matter. Now before the RS convention, stereochemistry was related to plus minus glyceraldehyde. So D-glyceraldehyde and L-glyceraldehyde, basically the D carbohydrates will have the OH group of the highest number chiral carbon pointing to the right in the Fischer projection, as in the R plus glyceraldehyde. So for carbohydrates, the convention is to arrange the Fischer projection with the carbonyl group at the top for aldoses and closest to the top for ketoses. So the carbons are numbered from top to bottom. Okay, that is basically the concept of naming for the RS convention in case of carbohydrates. Stereochemistry for the carbohydrates. Okay. So carbohydrates are designated as D or L according to the stereochemistry of the highest numbered chiral carbon of the Fischer projection. Okay, so if the hydroxyl group of the highest number chiral carbon is pointing to the right, the sugar is designated as D, dextro, and if the hydroxy group is pointing to the left, the sugar is designated as L, levo. Okay, so most naturally occurring carbohydrates are of the D configuration. Now the aldotetrosis. So glyceraldehyde is the simplest carbohydrate, C3-aldotriose. The next carbohydrate is aldotetrose. Okay, then we have aldopentose, 3 chiral carbons, 8 stereoisomers. Aldopentose, pentose means 5, so C5. Aldohexose will be C6, 4 chiral carbons, 16 stereoisomers. Okay. Now reducing sugars. Reducing sugars are nothing but monosaccharides with a carbonyl group that oxidizes to give a carboxylic acid. They undergo reaction with Benedict's reagent to give the corresponding carboxylic acid. It includes the monosaccharides glucose, galactose, and fructose. Now oxidation of D-glucose. So D-glucose glucose reducing sugar. So as we talked about in the Benedict's test, in the presence of Cu2 plus with the Benedict's reagent, it can give rise to D-gluconic acid, and plus Cu will be in the reduced form. So glucose is oxidized to carboxylic acid. So reducing sugar is basically any sugar that either has an aldehyde group or is capable of forming one in solution through isomerism. Okay, and reducing sugars can be oxidized by relatively mild oxidizing agents such as cupric ion, Cu2 plus. The carbonyl carbon of the aldehyde group is oxidized to carboxylic acid at the same time. Cupric ion is reduced to cuprous, Cu1 plus ion. Sugars with ketone groups, they are open chain former capable of isomerizing with via a series of tautomeric shifts to produce an aldehyde group in solution. Ketone bearing sugars like fructose are considered reducing sugars, but it is the isomer containing an aldehyde group which is reducing, since ketones cannot be oxidized without decomposition of the sugar. Under the basic conditions of the oxidation process, carbon 1 of a ketose is not oxidized directly. Instead, two ketones exist in equilibrium with an aldose by way of an enediol intermediate. If you can see, it's an enediol intermediate, keto form fructose. It's converted to the enediol form and then enol form, the glucose. A reduction of monosaccharides. So reduction of monosaccharides, it involves a carbonyl group, produces sugar alcohols such as alditols, and such as D-glucose gives D-glucosidol, also called sorbitol. Okay, so that is, if we come to oligosaccharides,
are carbohydrates formed of 2 to 10 monosaccharides units concurrently bonded to each other by glycosidic bonds. According to the number of sugar units, they are classified into disaccharides, and so on. The figure here represents the chemical structure of raffinose, which is a trisaccharide composed of galactose, glucose, and fructose. Raffinose can be found in peas, beans, cabbage, broccoli, other vegetables, and whole grains. So this is disaccharide. The disaccharides are oligosaccharides formed of 2 monosaccharides units covalently bonded by glycosidic bonds. The most physiologically important disaccharides are maltose, lactose, and sucrose. Hydroxyl group formed as a result of hemiacetal formation is highly reactive when compared to other hydroxyl groups. So this hydroxyl group present in one monosaccharide reacts with any one of the hydroxyl groups attached to C1, C2, C3, C4 or C6 of another monosaccharide to produce 1 to 1, 1 to 2, 1 to 3, 1 to 4 and 1 to 6 linked disaccharides. So only when one anomeric carbon is involved in glycosidic bond formation, reducing disaccharides are formed. If both anomeric carbon atoms of monosaccharides are involved in glycosidic bond formation, that results in the formation of a non-reducing disaccharide such as sucrose. In the case of reducing disaccharides, one end of the molecule having free anomeric carbon is called reducing end, and the other end where the anomeric carbon is involved in glycosidic bond is called as the non-reducing end. Then we come to maltose. Maltose is a disaccharide made up of 2 glucose residues joined by glycosidic linkage between C1 of one glucose residue and C4 of the other. So the configuration of the anomeric carbon of glucose involved in the linkage is alpha, and hence the glycosidic linkage is known as the alpha 1,4 glycosidic linkage. Okay. So the anomeric carbon of the second glucose is free. Okay. And therefore maltose is a reducing sugar. Second glucose residue is capable of existing in alpha or beta conformation, and maltose has been recorded occasionally in plants. It is usually obtained as a product of the enzyme hydrolysis of starch during germination or malting process. So also this is maltose, also known as malt sugar. It is used as a sweetener and as a substrate for fermentation. So it's a reducing sugar. The hemiacetal group on the right unit of D-glucopyranose is in equilibrium with the free aldehyde and can be oxidized to a carboxylic acid. Now lactose, also known as milk sugar, it's a principal sugar present in milk. It accounts for 5 to 8% of human milk and 4 to 6% of cow's milk. It's digested by the enzyme lactase. So pure lactose is found in whey, the water product, byproduct of cheese production. Okay. And it is made up of galactose at the non-reducing end and glucose at the reducing end. They are connected by beta 1,4 linkage. It has a free aldehyde group, so it is classified as reducing sugar. Then we come to sucrose, which is also known as table sugar. It is the most abundant disaccharide in the biological world. It is found in fruits, sugarcane and sugar beets. Caramel is a solid residue formed from heating sucrose. Some flavoring agent called invert sugar is produced by the hydrolysis of sucrose under acidic conditions, which breaks it apart into glucose and fructose. Invert sugar is sweeter than sucrose because of the fructose. Some of the sugar found in honey is formed in this fashion. Invert sugar is also produced in jams and jellies prepared from acid-containing fruits. Now in sucrose, carbon 1 of alpha D-glucopyranose bonds to carbon 2 of D-fructofuranose by an alpha 1 to glycosidic bond. Now the anomeric carbon atom of glucose C1 and fructose C2 are involved in linkage and is therefore a non-reducing disaccharide. Sucrose is a major intermediate product of photosynthesis, and it is the principal form in which sugar is transported from the leaves to other portions of plants via their vascular systems. Disaccharides: lactose hydrolyzed by lactase, maltose hydrolyzed by maltase, sucrose hydrolyzed by sucrase. All these enzymes found on surface of epithelial cells lining small intestine. So these are major complex biomolecules of cells. For example, the building block of protein will be amino acids, which is the basic structure and function of cell. Building block of DNA will be deoxyribonucleotide, which will basically function is hereditary information passing on that. RNA will be ribonucleotide. The main function will be protein synthesis. Polysaccharide is monosaccharide, the main function is storage form of energy. For lipids, it's fatty acids and glycerols, which main function is to meet long-term demands, storage form of energy. Okay. So carbohydrates are optically active polyhydroxyaldehydes, ketones, or the compounds which produce such units on hydrolysis. And how they can be classified, we have already discussed based on the number of units. Monosaccharides, simple sugars with multiple OH groups, disaccharides, oligosaccharides, polysaccharides. So according to the functional group present, aldose if the carbohydrate contains an aldehyde, aldotras containing 3 carbon atoms, aldotetrose, aldopentose, aldohexose, and ketose if the carbohydrate contains a ketone, ketotras, ketotetrose, ketopentose, ketohexose. So these are the monosaccharides. Aldoses have an aldehyde group at one end. Ketoses have a keto group usually at C2, if you can see over here. This keto group is at C2, D-fructose. We have also talked about D versus L designation are based on the configuration about the single symmetric C in glyceraldehyde. Okay, so these are the Fisher projections. These for sugars with more than one chiral center, D or L refers to the asymmetric carbon farthest from the aldehyde or keto group. Okay, and most naturally occurring sugars are D sugars. All those carbohydrates which reduce failing solution and tolerance region are referred to as reducing sugars. In disaccharides, if the reducing groups of monosaccharides, that is the aldehydic or ketonic groups, are bonded, these are non-reducing sugars. Example, glucose, sucrose. So on the other hand, sugars in which these functional groups are free are called reducing sugars. For example, maltose and lactose. This is a preparation of glucose. Structure of glucose, reaction, reagent products, obviously what all these reactions prove basically. So on prolonged heating with hydrogen iodide, HI, it forms N-hexane. Okay, it forms this. So this reaction basically proves that all the 6 carbon atoms are linked in a straight chain. For this case, glucose reacts with hydroxylamine to form an oxime and adds a molecule of hydrogen cyanide to give cyanohydrin. So these reactions basically confirm the presence of a carbonyl group in glucose. In here, glucose gets oxidized to 6-carbon carboxylic acid, gluconic acid on reaction with mild oxidizing agent like bromine water. So it is basically proves that the carbonyl group is present as an aldehyde group. Then acetylation of glucose with acetic anhydride gives glucose pentacetate, basically which confirms the presence of 5 OH groups attached to different carbon atoms. On oxidation with nitric acid, glucose as well as gluconic acid both yield dicarboxylic acid, saccharic acid. This indicates the presence of a primary alcoholic group in glucose. So pentoses and hexoses can cyclize as the ketone or the aldehyde reacts with the distal OH, and glucose forms an intramolecular hemiacetal as the C1 aldehyde and C5OH react to form a 6-member pyranose ring named after pyran, and these representations of the cyclic sugars are called the Howard projections. The cyclization of glucose produces a new asymmetric center at C1. The two stereoisomers are called anomers alpha and beta. So Howard projections represent the cyclic sugars as having essentially planar rings with the OH at the anomeric C1. So alpha will be the one having OH below the ring and beta will be the one having OH above the ring. Reactions and facts about glucose which cannot be explained by its open chain structure. So despite having the aldehyde group, glucose does not give Schiff's test and it does not form the hydrogen sulfide addition product with NaHSO3. The pentacetate of glucose does not react with hydroxylamine, indicating the absence of free CHO group, and glucose is found to exist in 2 different crystalline forms, which is alpha and beta. Alpha form of glucose is obtained by crystallization from concentrated solution of glucose at 303 Kelvin, while the beta form is obtained by crystallization from hot and saturated active solution at 371 Kelvin. So all these behaviors, this bulletin point behaviors cannot be explained by the open chain structure. Then if you move to fructose, fructose is an important ketohexose. It is obtained along with glucose by the hydrolysis of sucrose. Okay, so fructose forms a 5-member Frodano's ring by reaction of the C2 keto group with the OH on C5. So its two cyclic forms alpha and beta are obtained by the addition of the OH at C5 to the C double bond O group, and the ring thus formed is a 5-membered furanose ring. So the cyclic structures of two anomers of fructose are represented by the Howard structures over here as given. Okay. So this brings us to the end of this question. If you have any doubts regarding this question, you can ask me or we'll move on to the next question. Okay, we'll move on to the next question. Which of the following molecules is a component of in all of peptidoglycan, hyaluronic acid, and chitin? Okay, so these are the four options: N-acetylmuronic acid, glucuronic acid, N-acetylglucosamine, and acetyl galactosamine. I'll give you 2 minutes to think, and we can discuss the answer after that.
Okay, so the answer for this will be N-acetyl glucosamine. We'll discuss the it. So we've already talked about the basic thing about carbohydrates, basically polyhydroxyacetones or aldehydes or polyhydroxyketones. We know about the biological roles, nutrients and energy, storage, structural and protection roles, bones, lubricants, cellular adhesion, all of that. So we know about the group classification, we know about the structure and chemistry, aldose, ketos. Okay, we know about the monosaccharides, aldoses, aldotriose, aldotetros, aldopentose, aldohexose. We know about the ketoses, dihydroxyacetone, D-erythrulose, D-ribulose, D-xylulose, D-fructose, D-sorbose, D all of this. So what is the chiral center? All of these questions are there based on the sugars. So we need to figure out the answers to all these questions. They talked about monosaccharides, which are reducing sugars with free anomeric carbon atoms that are reasonably good reducing agents and will reduce hydrogen peroxide, ferricyanide, or certain metals such as copper. There is a variety of chemical and enzymatic reactions that produce derivatives of the simple sugars. Reductions like aldehydes and deoxy sugars. The carbonyl group is reduced to alcohol. Okay. Then we have reductions aldehydes and deoxy sugars. Deoxy sugars, one or more hydroxyl groups are replaced by hydrogens. Amino sugars at the C2 position. The amino groups can accept an acetyl group. The final products are sugar derivatives with important biological roles, such as beta-D-glucosamine, beta-D-galactosamine, beta-D-glucosamine, N-acetyl. See, N-acetyl, beta-D-glucosamine, muramic acid, N-acetyl muramic acid. So oxidation at the aldoses CN. We know that D-glucose can give oxidation at C1 will give rise to aldonic acids. Oxidation at C6 will give rise to uronic acid, and oxidation at C1 and C6 will give rise to aldaric acids. Okay, you can see oxidation at C1, COH at the top, aldonic. Oxidation at C6, COH at the bottom, uronic. Oxidation at both the ends, 1 and 6, aldaric. Sugar esters, phosphate and sulfate ester of monosaccharides. Okay, these are the sugar derivatives. Another type of sugar derivative, which is sugar ester. O-glycosidic bond. So two monosaccharide units are linked by a covalent bond, alpha glucose, alpha D glucose, and beta D glucose. Okay, so hydrolysis and condensation. So that will give rise to alpha D glucopyranosyl 1,4-D glucopyranose. Maltose is a component of malt, reducing disaccharide. Okay, so all oligosaccharides are components of milk will be reducing disaccharide. Sucrose is a component of many higher plants, no free anomeric carbon, non-reducing disaccharide. Polysaccharides is homopolysaccharides, heteropolysaccharides. We know about this linear chain and branch chain. Then heteropolysaccharides can also be of linear or branch chain. And polysaccharides can contain two polymers of glucose, amylose linear chain, amylopectin branch chain. Okay. So that's polysaccharide, homopolysaccharide starch. The enzyme alpha amylase catalyzes the alpha 1,4 bonds digestion. Polysaccharides, homopolysaccharides, glycogen. The storage polysaccharide in animals. It is stored in the liver and skeletal muscle. Its structure is similar to amylopectin structure but with higher branch points, high compact structure. So polysaccharides is structural polysaccharide in plants. The linear chains can be constituted by D-glucose units linked. No hydrolyzable by human being enzymes. Hydrolyzable by cellulases. Okay, insoluble in water, fibrous and resistant, and cellulose chains interact by means of hydrogen bonds forming cellulose microfibers, and the hydrogen bonds increase strength of the structure. Okay, so structural differences due to the nature of the bond, alpha 1,4 and beta 1,4. So this is amylose helical conformation, or this is cellulose. Okay, so structural polysaccharides can be present in cell walls of fungi and the exoskeleton of crustaceans, insects, and spiders. The linear chain is constituted by N-acetyl D-glucosamines in beta 1,4 linkage.
Okay. Now, heteropolysaccharides, such as peptidoglycan, is a component of the bacterial cell wall. It is a one-fold link polymer of N-acetyl glucosamine and N-acetyl muramic acid units, and this polysaccharide is joined to a tetrapeptide by a covalent bond. This lysosome can basically hydrolyze these glycosidic bonds. So they are present in animal cells. Glycosaminoglycans are present in animal cells, which we call GAG or GAGs. The GAG are linear polymers constituted by disaccharides. One of the monosaccharides is always N-acetyl glucosamine or N-acetyl galactosamine. The other one is usually uronic acid. And they are negatively charged sulfate or carboxylate groups. They promote molecular associations, charge-charge interactions, and they can recognize specific ligand cell interactions. Then we have hyaluronic acid. It is not protein linked. It is highly hydrated by virtue of strong interactions between water molecules and the polyanionic complex. It is present in cartilage and tendon, vitreous humor in eye, HCF on the extracellular matrix, mucosal surface and synovial fluid. Or, for example, in case of chondroitin sulfate, it promotes tension resistance in cartilage, tendon, and arteries. It is present in brain, kidneys, and lungs. Dermatin sulfate, which is present in skin, veins, and cardiac valves. Its concentration increases when cells grow old. Hyaluronic acid is basically not protein linked. For keratin sulfate, we have this minor component of the proteoglycans is present in bones, cartilage, horns, and intervertebral discs. And heparin, natural anticoagulant substance, is present in mastocytes. It contains D-glucuronic acid. So these are the where GHGs are present. Hyaluronate, chondroitin 4 sulfate, chondroitin 6 sulfate, heparin, keratin sulfate, dermatin sulfate. So you see, hyaluronate also has N-acetyl glucosamine. So complexes of protein or lipids with oligosaccharides and polysaccharides covalently linked are present in the cellular surface and in the extracellular matrix. They are positive side of the cytoplasmic membrane orientation. Okay, so proteoglycans will proteins linked to the polysaccharides GAG. Glycoproteins will proteins covalently linked to oligosaccharides. And glycolipids will membrane lipids and sphingolipids covalently linked to oligosaccharides. So nucleus protein is covalently linked to one or more than one GAG. And proteins and carbohydrates are linked by means of N or O glycosidic bonds. So glycoprotein structure is more complex than proteoglycan structure. Structure of the oligosaccharide is very important in the identification of the protein to interact with. Localization in positive side of the cytoplasmic membrane will be glycophorin A in erythrocytes, proteins secreted by eukaryotic cells, immunoglobulins, hormones, coagulation factors, proteins from lysosomes. What are the biological advantages? The polarity and the solubility of a protein can be modified. They can identify the final location of just synthesized protein, protection against enzyme digestion, and specific biological roles. For lectins, this is a class of proteins that can bind to carbohydrates with high specificity and affinity. They participate in several processes such as cell signaling and adhesion. They also control the intracellular location of the proteins that are just synthesized. In animals, the contact between cells interactions. Physiological roles such as leukocyte movement, microorganisms, infection, and toxic mechanisms. Okay. So, if you have any, that we come to the end of this question, discussing this question. If you have any more questions related to this, whatever we have discussed today, you can ask me, or we can stop the session for today. Does anybody have any questions regarding whatever we discussed today? Okay, thanks. So if nobody has any question regarding whatever we discussed today, maybe we can stop the session. I'll upload the recording and I'll upload the slides also. If you ever have a question later also, we can have a discussion and we can conclude the session for today, and we can have the next session next Monday at 6 p.m. Okay. So I'll stop sharing the screen. Thank you so much. Thank you. I'll stop sharing the screen.